We're very fortunate today to have a visiting professor joining us from the University of Vermont. And I'd like to introduce Dr. Jeffrey Spees. He's professor of medicine and neuroscience and one of the premier scientists in this field, which spans cardiology, neurosurgery, neurology, and basic science. He comes to us really because of the fact that Dr. Wesley worked with him earlier in her career before she came here, as a great example of a basic scientist. Both are a basic scientist that can work well with the clinical problems. And we've asked him to talk today a little bit about some of his research on the conditions which would be important for brain injury and brain repair. This comes from looking at the basic science of everything from small molecules like we do, or like genomics like Dr. Elish does, or last night our discussion with Dr. Schuster about some of his work on blood-brain barrier. These are basic scientists that are really working with the basic questions which underlie all the diseases we treat and will be the basis of how we will improve care into the future. So it's a delight to have you, Jeffrey, and thanks very much for coming to join us.
Can you hear me? Yep. So I just wanted to say thank you to Wesley and also Dr. Dempsey. It's great to come out here. It's only really two of us working on stroke at UVM. So to see like the Department of Neurosurgery collaborating with bioengineers and cell biologists and all working to treat stroke is really inspiring. So my lab works on glial cells, astrocytes and microglial cells, which are really important in regulating the stroke injury environment. In my talk, I don't have anything to disclose in terms of, we are working on biologics and things to treat stroke, but I won't talk about them. The goals for this talk are to describe the prevalence of cerebral ischemia and some risk factors for stroke, which many of you are probably familiar with. Describe the location and phenotypic markers for reactive astrocytes in stroke, explain some of the functional roles of reactive glial cells at different stages of stroke and recovery, and then really to kind of focus on some of our work looking at paracrine and juxtacrine aspects of cell signaling in the stroke peri-infarct area. So by paracrine, that would be like one cell at a distance from another cell secreting a hormone or a growth factor that hits a receptor on a distant cell. Juxtacrine, the cells are next to each other with special kinds of receptors and ligands that are locked on the cell membrane. So I'll show you some of that too. As you know, stroke is really prevalent. There's almost 10 million people that have had stroke in America. And the new and recurrent attacks are increasing every day. And the mortality and morbidity are a real burden on the healthcare system. And so here you can see the projected cost of cardiovascular disease and stroke going out to 2035. Stroke is here. And so the projected cost of stroke this year is about $91 billion. And by 2035, it's projected to be almost $150 billion a year. The total cost of cardiovascular disease, including coronary artery disease and stroke and hypertension, almost $1 trillion we're gonna hit. So it's gonna be a real problem for patients and for economics. Excuse me. There's many risk factors for stroke. I won't go through all of them. You're familiar with hypertension and diabetes and hyperlipidemia and obesity. Some of the things, if you're interested in this, the AHA puts out a really great report, and the 23 report is especially good, looking at all risk factors for cardiovascular disease. Some interesting things I found, even low use cigarette smoking. So one cigarette a day as opposed to 20 or more cigarettes a day, that doubles your chance of having a stroke relative to 20. So you don't have to smoke very much. So I encourage you not to smoke at all. Sleep apnea severe obstructive sleep apnea doubled the risk of stroke, but mild or moderate sleep apnea did not. And so this may have to do with hypertension induced by the sleep apnea. Prior history of disease, such as transient ischemic attacks, or the combination of heart disease. So if you have enlarged chambers in your heart or atrial fibrillation, it increases your chances of stroke, and also kidney and liver disease. Of course, sex, so females have a higher chance of having a stroke than males, and about 55,000 more strokes happen this year in females than males. Also, race, so especially in the South, the Black African American population and Hispanic population have typically more strokes than Caucasian population. And after talking to Dr. Dempsey last night, the Native American population may even be more at risk of stroke than the African American population. And finally, increasing age increases your risk for stroke. Some of the really exciting developments since I started working. So I started working in stroke in about 2005. And in 2015, a series of trials, the Mr. Clean and Extend, Revastat, a series of clinical trials using stent retrievers, which many of you probably use on a daily basis, completely changed the treatment of stroke. And so now the kinds of considerations are like the time of onset of stroke, of course, but also, do you have a CT scanner? And do you need to transport to a center like this that has scanning capability? Because the combination of neuroimaging with stent retriever technology really revolutionized the treatment of stroke. And now maybe 80% of patients are having embolectomies, as opposed to just maybe getting TPA or getting aspirin and going home. So the other thing that is really exciting is that the time window to treat stroke has changed dramatically. So in about 4.5 hours, you could treat somebody with a tissue plasminogen activator like Alteplase. However, after 3 hours or so, the advantages of using a thrombolytic really go down. And so the exciting part is this mechanical embolectomy within 0 to 6 hours. Almost all patients are getting this now through a femoral or radial access. And in fact, you could even perform that coincident with TPA infusion. And some patients now, later trials, and maybe around 2018 or 2019, showed out to 24 hours patients were benefiting from receiving embolectomy. And so not all patients, of course, depending on the size of the lesion and the vessel that's involved and the risk of intracerebral hemorrhage, you may or may not choose to do this out to 24 hours. But there could be a time when almost all patients receive embolectomy, which is exciting. And then finally, things may start to shift from use of Alteplase to tenecteplase, like it did in cardiology, where you can have a single bolus rather than like a 60-minute infusion that you have to deal with. And finally, the patients are still receiving aspirin or an antiplatelet like clopidogrel, and people are often left, rather than trying to treat their hypertension, it's often better to leave them with a little blood pressure to keep them perfused. Many of you are familiar with all the changes in imaging that have occurred. So patients will get non-contrast and some will get contrast CT scans. In some cases, they'll even have like a perfusion study done. But the really interesting stuff is with the imaging of the vessels now. You can actually, here in blue, you can image well-perfused vessels, low perfusion or badly perfused vessels, and even collaterals here in blue. And all of those can help inform a decision to triage or treat. Also, the advances in software are really exciting. So the ability of AI in some cases to help determine clinical decisions here is really encouraging. So I think that we'll see a lot more of this kind of help with AI and imaging and decision whether to use thrombectomy or not. And so in my lab, when I started in stroke, most patients weren't, some were receiving thrombectomy, but most were not being reperfused. And so the situation has totally changed. And now we're adopting the reperfusion model of stroke in my lab. But the studies I'll show you today are with a permanent ligation model where we go through a small hole in the skull and we ligate the middle cerebral artery. And in fact, we do this with like an ophthalmic Tenno suture, like a spider web suture under microscope. And we often will break the vessel distal to the ligature just to make sure. So if the vessel's not ligated well, the animal will bleed out, and so it won't get into our study. So this ensures a very reproducible focal cortical infarct that we can study astrogliosis in. And so this is kind of a map. We mainly focus on this peri-infarct area, which is surrounding the infarct. And the reason we do that is because the astrocytes there have a lot of potential to treat stroke in terms of their functions and the things they secrete. And basically, if we can kind of learn what they do and provide that at an earlier time point, because it takes maybe 6 to 12 hours for some of these factors to be produced and secreted. So if we know what they are and we can deliver them at the time of reperfusion, it could really change the treatment paradigm. So when we're studying reactive astrogliosis, this is a stain for gliofibrillary acidic protein, which is an intermediate filament protein. It's a cytoskeletal protein in the astrocytes that we can track them with. And often when there's an injury, depending on the size of the injury, the cells will undergo hypertrophy, and the ones closest to the injury will proliferate. And those proliferating cells have different functions than the astrocytes that are back from the injury. In terms of regulatory functions of astrocytes, they're really important in terms of regulating, for example, neurovascular coupling. So hopefully the talk I'm giving right now is requiring some thinking on your part, and it's requiring some nutrients from your blood to be available for your neurons to fire. And the astrocytes have end feet around the blood vessels, and they're regulating the synapses of the neurons. So they actually kind of mediate the amount of energy and nutrients that neurons are getting to respond to things. And in addition, they form the structural microarchitecture of the brain. So it's kind of like the scaffold that everything is built upon. And they control water availability. They regulate the blood-brain barrier through their end feet and interactions with endothelial cells. And they also regulate immune cell responses, which I'll show you a little bit, and gliotransmission. So the astrocytes are actually linked and interacting electrically and through calcium signaling to regulate kind of large-scale firing of neurons in the brain. Finally, they're regulating synaptic transmission directly by regulating the synaptic cleft. Not sure where that is. So astrocytes were like the first thing that really happens if there's a leak in the vasculature, the microglia, which is a blood-derived cell that in development, microglia migrate from the blood system into the brain and then become trapped in the brain. And they live there and they act as sentinels for injury. They also clean up the mess by phagocytosis after the injury. And those cells initially will receive a signal like endothelin or reactive oxygen species or thrombin or something that's released, fibrin that's released from the vessels, and then they signal to the astrocytes. And there's a crosstalk between microglia and astrocyte that regulate how the brain responds to the injury. And so initially astrocytes will respond to microglia by undergoing hypertrophy and proliferating. And later on in the injury, as it resolves, they'll form an astroglial scar, which is initially thought to be, it was seen as kind of negative because it was believed that blocked the ability of neural axons to repair injury, but now it's known that's not the case. They also regulate the release of extracellular glutamate. And finally, they need to reestablish the blood-brain barrier, because if they don't, the immune cells from the periphery will continue coming into the brain and the injury will get larger and larger. So my lab studies cell signaling, and the basic goal is to understand better the signaling of the astrocytes in the peri-infarct area so we can develop drugs to treat stroke injury. And so one of the systems we study is called NOT signaling, and this is an example of juxtacrine signaling, where there's a ligand, Jagged 1, on one cell, and it's kind of, instead of throwing the football pass, that would be a paracrine response, this is more of a handoff. So the ligand, there's actually mechanical tension required between the ligand that's on the surface of one cell, that's actually more like a receptor, and the receptor on this cell. And when these tug on each other, there's a cleavage step that occurs from an enzyme complex called gamma secretase. And that little intracellular domain that's cleaved from the cell then goes into the nucleus, removes a co-repressor complex, and starts gene transcription for a whole variety of genes to respond to the injury. And so this system is really important in astrocytes and neural stem cells because it regulates their decision to self-renew, for example, in the case of stem cells. It controls their identity by controlling the genes that are expressed by the cells, and it also controls their proliferation, which is something that we've shown. So gamma secretase that I mentioned that cleaves the little intracellular domain, we have inhibitors for that. So for example, DBZ, shown here, diaminobenzidine, we can, if you inject that into the brain in a direct kind of stab injury, you can actually see, these are control astrocytes that got vehicle, you can actually see that it affects the morphology of the astrocytes, and we can quantify that. And in addition, after stroke, so this line shows you the demarcation of the infarct and peri-infarct area. This is GFAP staining to show you the astrocytes. Here's a blood vessel. Here's an astrocyte with an end foot on the blood vessel. And so if we stain for these intracellular transcription factors like NICD as the Notch intracellular domain, you can see that almost all the astrocytes in the peri-infarct area, very close to the infarct, are expressing in their nucleus as the green stain here, this NICD. And my lab showed that this marker RC2, actually, this is a lot of data summarized here in this pie chart, like many animals with strokes were quantified. And by staining for a proliferation marker, Ki67, shown here in the nucleus, and reactive astrocyte markers, we can quantify the number of reactive astrocytes that are proliferating. And so this RC2 tags about 73% of the proliferating reactive astrocytes. If we treat stroke animals with this gamma secretase inhibitor DBZ, we blunt the proliferation of the reactive astrocytes because we're inhibiting Notch and some other cleavage products. And then to be more specific, because you can have off-target drug effects of DBZ, we design these transgenic mice. And these are fancy mice where the mice develop normally. You breed these two mice to each other, first of all. One has a GFAP promoter, the promoter for that, the gene of that protein I showed you that stains the astrocytes. And then you have another mouse that has the receptor Notch1 floxed by these LOXP sites. And basically, if you breed these to each other, the mice develop totally normally, and they don't know that there's anything different about them than a normal mice, and you couldn't tell either. However, if you inject them with tamoxifen, based on this element, it causes Cre-recombinase, a special enzyme, to go into the nucleus, and it will target these LOXP sites and cleave out Notch1, the gene for Notch1. And so this system allows you with really good fidelity to treat animals with tamoxifen, then you can wait a few weeks to a month, and all the Notch1 receptors, maybe 80% of them, will be deleted from the astrocytes in the brain, and they're just sitting there waiting. And then you can do the stroke model and see what happens to the astrocytes that are missing that receptor. And we were able to show that, similar to the gamma secretase inhibitor study, that there's a significant reduction in the astrogliosis that occurs after stroke. So we now know that Notch1 regulates the proliferation of the astrocytes in that peri-infarct area. And these are the astrocytes that secrete a whole variety of neuroprotective compounds and other important things. And another thing we found, this is standing for CD45, which is like a pan blood cell marker. If we treat either with DBZ or with the Notch model I showed you, green is the CD45. Normally, there's breakdown in the blood-brain barrier, and from the infarct core, immune cells spill into the injury, which causes this kind of liquefaction injury that gets worse and worse as time goes on. And so that the astrocytes form this kind of barrier to that. So these astrocytes proliferate, they release factors, and they actually prevent the immune cells from migrating into the brain deeper. And when we treat with DBZ, we see that that kind of wall is broken and the immune cells start to migrate into the injury. And based on the immune cells migrating in and other factors, we see after gamma secretase inhibitor treatment that the size of stroke gets larger. And similarly, if we treat for, we give 3 injections of tamoxifen, we wait 28 days in this special transgenic model, we can see that at day 3 after stroke, there's a significant increase in infarct size. So that tells us basically that this Notch signaling is really important in the peri-infarct area and gives us kind of a target to work with. And then we also looked at Jagged1, which is the ligand for juxtacrine signaling. So the one cell has a Notch receptor, the other cell has the Jagged ligand. And we found that there's subpopulations of cells going out from the peri-infarct area in terms of like cells that are directly adjacent to the injury versus distal to the injury. And these cells closest to the injury express high levels of Jagged1, and they also express this endothelin receptor B that I'll tell you a little bit about, a G protein coupled receptor. And this signaling system is a paracrine system where there's a 21 amino acid hormone endothelin that's a potent vasoconstrictor. There's 2 G protein coupled receptors, A and B. When it signals through A, endothelin causes vasoconstriction. When it signals through B, it often causes vasodilation and a whole bunch of other things that we're studying now. And so using this system, we're able to map, using fluorescent activated cell sorting, these populations. So if you're looking at a control animal that is treated with oil and not tamoxifen, oil is just the tamoxifen gets its lipid, it's basically dissolved in oil to deliver it. So in a control animal, there's these populations of cells that are Jagged negative or Jagged low. Then when you have a stroke, on the contralateral side, you don't have these cells appear. On the ipsilateral side, you have those Jagged high endothelin receptor B positive cells appear. And then we found that if we treat with tamoxifen, here's that population, we ablate that population from the brain. And that, we think, is why those cells, the immune cells, are migrating in. And finally, to look further at this endothelin receptor B, the G protein coupled receptor, we created similar mice where the EDNRB is the gene for endothelin receptor B, and we knocked that out. And then we found that when you treat the animals with tamoxifen, similar to Notch, it's a phenocopy. So the Notch we found is regulated through it's regulating STAT3, a transcription factor, which is then regulating endothelin receptor B. And if you remove endothelin receptor B, you get a very similar kind of effect where you remove the reactive astrocytes that are proliferating. So in summary, I think it's really exciting and inspiring, the developments that you guys are working with the stent retrievers. It's really kind of brought stroke into the same kind of vein as interventional cardiology. And I think now it's up to people like me and other researchers to work out these factors, because many clinical trials were done with neuroprotectants that did not work out. And so part of the problem is we need drugs that modulate immune cell responses, like I showed you, that protect and preserve and repair the blood-brain barrier and also neuroprotect simultaneously. And that's why just treating with something like tacrolimus doesn't work. And so we really need kind of combinatorial studies now to look at pharmacologics and biologics. Or, for example, like Dr. Vegamandi working on, you could potentially use non-coding RNA or a mimic, which targets multiple factors simultaneously, maybe even hundreds of genes, and that may provide an avenue to treat stroke as well. And so, in conclusion, the identification of targets downstream of Notch and endothelin receptor B signaling may help inform the discovery of new biologics to treat stroke, and I'll talk a little bit about that in the next talk that I'll give today at lunch. So again, I want to thank Dr. Dempsey and Dr. Wesley for inviting me, and I want to acknowledge the NIH for funding our studies for 18 years and a whole bunch of members in my lab that helped with these studies. Thank you.
Thank you. That's wonderful. I'm really grateful for you to very clearly put glial cells and the future microglial cells on the forefront of our imaginations here, because quite frankly, we've used them as markers of disease in oncology and as a black box. Everybody concentrates on vessels and neurons, and to understand the regulatory processes which determine health and disease is really what you're doing, which is fantastic. Do we have questions for Dr. Spees? Especially from our scientists who have taken the most remote corners of the room, I notice.
It's a great.
I am.
Wondering, you are knocking down notch 1, there are other notches as well. Do you see any compensatory effect of the other notch?
That's a good question. I think in some cases, especially with other cell types, we could. It's hard to say with astrocytes, because when we remove notch 1, those astrocytes that we're studying are gone. They proliferate, and if they don't proliferate, they're gone. And so I do think, like, for example, notch 3, right, which was shown to be involved in smooth muscle cells and cadacil, which is a vascular disease that leads to stroke. I do think we need to go now and look at notch 2 and notch 3, especially in astrocytes, because I do think they're important, probably for other functions.
Repairing astrocytes is a great strategy, but many people think that you have to treat the whole neurovascular unit rather than just the astrocytes to get any recovery of the stroke, like pericytes or endothelin. So I just want to know your thoughts about it.
Yeah, I certainly think it would be advantageous to study everything at once. Our models are so precise that we could potentially miss something. So that is one of the reasons that if you do take a ligand like Jagged, you're not going to just treat astrocytes, right? If you inject a soluble version of Jagged, you're going to treat endothelial cells. You could, it just takes empirical studies like you're doing to know what it will do to the other cells. But this is useful because this is a system where we can really control it. And now we're actually able to do screens where we knock out particular receptors and then see what's the result in terms of what the cells secrete, and then it gives us a bank of things to test for treatments. So I agree that it's important to consider holistically the neurovascular unit.
One of the things that we think of a lot is that these processes are not just a response to injury, but an initiation of repair. Where do you think the glial cells fit in this area of injury as far as being a target for progenitor cells to migrate to, or a secretor of substances which might initiate such a process?
So the talk I'll do next, which we're really focused now on the communication between the microglia and the astrocytes, because it starts off, the microglia talk to the astrocytes and get them all revved up and keep immune cells from coming in from the periphery. But then the astrocytes talk back to the microglial cells that can change their phenotype. And there's this kind of crosstalk that goes on for weeks, really. And so, and as you know, eventually the immune system often becomes too repressed, and then patients are opened up to other complications. But yeah, I think so. It's this crosstalk and communication that we need to study for, and even for example, the signals that tell the astroglial scar, like if the astrocytes don't proliferate, you'll have intracerebral hemorrhage. If they overproliferate, you'll have a glioblastoma. And so there has to be really tight controls and communication between these different cell types. And that's why some of the signals are juxtacrine in nature. So it like you said, you don't want some of the signals to go out, like EGF, for example, and stimulate the wrong cell type. You need some of them to be either lipid soluble or juxtacrine in nature, so that they stay tight and the communication stays tight.
This is a really important point for the clinicians. We, as clinicians, tend to be very black and white about things and say fever's bad, edema's bad, kill the fever, kill the edema, whereas it's probably part of a process of repair, injury, removal of debris, etc. And so much of what we are trying to achieve is to basically question authority, your professors who said, wipe out all edema, and rather try to maintain a homeostasis in your system. This is why I think we went overboard in some of our ICU treatments. When we really want to do is maximize cardiac function. Instead, we sort of drown people in fluids, things of that nature. This is really what we're doing here at a cellular level, is trying to find what is the mechanisms of health and disease. And that's one of the reasons why we combined here the oncology and the stroke labs journal clubs, so that they would begin to say, we're studying the same factors. That too much, too little at the wrong time can be a big problem, which is why I'm excited about microglia, because I think that they really are going to unlock a role. They've just been so hard to study. And Kip.
So kind of on that note of complication, Jody Waters here just actually gave a talk and was talking about macrophages once the blood-brain barrier has been violated, and then they enter the brain, but then adopt the glial cell phenotype, and then how that kind of plays into the communication. I'm kind of wondering how you're considering that in terms of the different stages and the macrophage transition.
That's a really great point. So I do think there's a lot of transdifferentiation kinds of things that are going on that people aren't studying well, because we don't have very good models for lineage tracing. And part of the problem is a lot of these cells share receptors and signaling factors. So like an example is like hepatocyte growth factor was discovered from the liver, but it affects like almost every cell in your body. So in the case of the brain, there is this kind of transition in I guess it's hard to for me to describe actually. You need, it's like Dr. Dempsey said, you need the immune system. Without it, you have a real mess. And it's like in cardiology. If you have too much fibrosis after an MI, you're going to have a stiff heart and you're going to have chronic heart failure. If you don't have enough fibrosis in the ventricle wall, you're going to have ventricular rupture. So it's this kind of fine line between like too much collagen and scarring or not having enough. And in the case of the microglia, I think it's really becoming apparent that this interaction between the microglia and the astrocytes in the case of like vascular dementia, for example, is going to be really critical to understand the kind of the early etiology of disease and the things that are occurring long before the storage problems actually occur. And of course, I didn't mention it, but the amyloid precursor protein pathway is also regulated by gamma secretase, and it's cleaved just like Notch. And so all these things are happening at the same time. So it gets pretty complicated. I don't know if I answer your question though. Yeah,
It's a hard question to answer.
yeah, it's a good question.
Yeah, but I appreciate the ability of this idea of a forum of people who can ask questions. And my hope is that the residents who have gone mute are spending all their time trying to think about what this would mean as they think about research projects, which affect, believe me, if you're studying the brain, this affects whatever condition you're studying. I don't care if it's functional, cerebrovascular, oncologic, traumatic, etc. It all comes down to what happens at this cellular level, and then the genomic level and the proteins that they produce. This is great. I want to also point out that, because I appreciate that a lot of us got to run off to take care of patients, it gives me an opportunity to say a few things about the visiting professor process. And unbeknownst to lots of people, the Landis family has been kind enough to support our visiting professor program. And this is a family of a grateful patient who lived for 20 some years after his initial surgery with us and died of other causes. And the family continues this because they're very interested in continuing our education. And so the sponsors of this are the Landis family. And it's allowed us to bring visiting professors. And we, as COVID clears, will be doing more and more of this on every topic. And for that, we're very grateful. We just want to show our appreciation to Jeff, who's really here as a visiting professor. He'll be here for really a little under 2 days. He'll be giving a noon lecture in the Jagged room, which means food, of course. But it'll be much more scientific, targeting at a lot of our scientists and the technologists and the students in the Ph.D. programs. All are welcome, and be meeting with various faculty and interested scientists throughout the time. We really appreciate you coming. It's just outstanding. We have a small token that allows you to remember Wisconsin. And thanks to the Landis family, we also have a check which will be sent to you as well.
Thank you.
So it's our pleasure. Thanks so much. So appreciate everybody being here today, and Jeff, appreciate your work. Thanks all.